Hello. We'll talk today about a technique for nucleic acid protein identification, which will show how to show the in vivo analysis of any interaction between a DNA and the protein, and especially with an epigenetic modified protein. So what's these epigenetics? Epigenetics, it's Bruce Lipton said that what you're thinking is translated into chemistry that will determine what you become. So basically, anything you do, anything you have, anything you even in behavior, it's all translated into chemistry and hence into genes. So we'll take a brief introduction. So and after that we'll deal with some of the work about how we can develop a small molecule inhibitor by using a technique of protein microarrays. Then we'll talk about another approach to detect how epigenetic modification on chromatin on phosphorylation on histone 3, and that's the histone modification. So basically, both these topics, subtopics, talk about the epigenetics and especially histone modifications. So what is epigenetic regulation? So normally, as you know, DNA is compacted around the histone, wrapped around histone to form a bead on a string structure that soon, upon higher level of compaction, will lead to a chromosome. So the histone itself is composed of nucleosome, where the histone is wrapped around a core histone composed of 8 histones, double histone of each of H2A, H2A, H2B, H3, H4. From each one of the histone, so we have 8 proteins here inside the core. From each one of these core histone, the amino acid sequence have a tail of amino acids. We call it histone tails. So you can see the structure of the nucleosome and connected to each other by a DNA. So generally, histones, as we said, it's 8 proteins, so we call it core histones. Each histone have a tail, and each tail can undergo modification on a specific residues over the tails. So you can see methylation, phosphorylation, acetylation, phosphorylation again, okay? And as well, there is another histone, which is called the H1 histone. And normally H1 histone stays on the entry and the exit of the DNA over the nucleosome. So it's kind of like wrapping the entry and the exit of the DNA. So normally it's existing here to bind it firmly and keep the structure of the nucleosome. So as a summary for this slide, you can see how these modifications differ based on the histone tail. You can see that the modification differs of histone 2A, then histone 3, then histone 4, then histone 2B. And all these modifications or epigenetic modifications control the way the gene is expressed. Okay, it's another simplified figure. You can see that histone 2A have 2 tails, as well histone 2B, while histone 3, you can see the tail of histone 3 and the tail of histone 4. And there is specific modifications on a specific amino acids. And these scientists found that these modifications are conserved. Numbers here denote the sequence of the amino acid. So this protein, the histone 2A, start with this amino acid, okay? So number 5 means it is the 5th amino acid in the chain in the polypeptide chain of H2B. So it's a matter, as I said before, this modification control the way of expression of the gene associated with these regions of histone. So you can see that within a number of modification, a gene can be opened and expressed while other modification can lead to shutting down the gene and it's not expressed. And if we know the perfect combination of these modification on the specific amino acid, we can open and close a gene for expression. So that's why scientists agreed to use a expression which is called histone code. So if you manage to solve this code, you can know when and where on the DNA you can open the DNA for expression for a certain gene or not, or close another one. So transcription of this genetic information can be regulated by chemical modification, as we said, methylation, acetylation, phosphorylation to histone proteins. Let's say, for example, or the consequence of these modifications. So other protein, when they recognize this modification, this specific modification, they will bind to this modification. For example, you can see a protein that have two domains. It's a single protein in the nucleus that have two domains. And the other protein, which actually the first subtopic we'll talk about it, it's called the Spinone. This protein have two domains, domain 1 and domain 2. And the domain 2 will mark it, it's named Tudor domain. And we'll see how these domains tend to bind to a modification on the histone proteins. Generally, there are three proteins, generally, either writers or erasers or readers. So basically, the writers actually, these are the proteins responsible for adding the signal over the histone protein. So these writer proteins, either acetylases adding acetyl group, or methylases added methyl group, or phosphorylases added a phosphate group. So a phosphate here, in order to be added to this serine amino acid, it needs a writer. And as well, there is erasers. Actually, they are the deacetylases and demethylases and these and the phosphatases, of course. Aside from these one, the writers and the erasers, there are readers. So readers actually read the signal and they do a specific job on the DNA, promoting either transcription or closing down the DNA based on the existence of the signal. And these readers, there are a lot of readers, including bromodomain, chromodomains, PhD finger, and as well Tudor domain. So basically, the first info you knew now that Spinone considered to be a reader. So let's see how the process is very complex. So you can see here an example for a histone, and the histone 3, and the histone, actually histone tail is exposed, showing all the possible modifications that may occur on a different amino acids. You can see that there is a methylation on arginine 2, there is acetylation on lysine 4, and this lysine as well can undergo methylation. You can see a phosphorylation on serine 28. That doesn't mean that histone 3 and of course other histones have to be having all this set of modification at the same time. It's a kind of combination between this modification to have a response on the DNA. So you have now a writer that put the signal over the amino acids, and you need a reader. So these are the different readers. So you can see chromodomain, 1433 domain, BD domain, Tudor domain, and of course these domains are specific for the signal. That means a Tudor domain, which we'll take this in this lecture, Tudor domain binds only to methyl arginine. So Tudor domain binds only to methyl arginine, as well as BD domain binds only to acetylated lysine. Okay, so you can see, for example, here, let's take for example this one. the acetylated lysine on the position 4 in histone tail can be methylated and can be acetylated as well. But once any of these domains binds to the methylated lysine 4, it will promote the expression or the block or the switching off of a certain genes. While if the BD bind to the acetylated version of this lysine, it will promote the regulation of other genes. So that's mean along the DNA, you can see histone 3, for example, at all the nucleosomes. But at some region, this histone 3 could be methylated, and at some other regions, this histone 3 could be acetylated. So it's a mode of regulation of the gene expression, and that's why it's called epigenetics, because now genes is not controlling. It's a force above the gene that control the expression of the genes, like the signal, like the readers. Let's talk how these small molecule inhibitor have been developed using a protein microarray to inhibit one of the important readers, which have a tutor domain, and as you know now, it recognizes the methylated lysine. So generally, to understand the idea, normally, spin1 protein, which is a reader, with its tutor domain, will bind to methylated lysine 4, and only methylated lysine 4 of histone 3. So the aim to understand the protocol and how we manage to understand or to experiment the interaction between a reader and the modification on histone, we need to develop an inhibitor that blocks spin1 to this methylated lysine 4. So that's the aim of this subtopic today. So what is spin1 anyway? Spin1 is a histone code reader, okay, and recognize only and binds to histone 3K4 trimethylated. So you need to understand now that it needs to bind to the trimethylated. It's not only one methyl signal, it's a trimethyl signal on lysine 4 of histone 3. This spin1, as you know now, it's a tutor domain containing protein. And one of the major functions of spin1 is activator of Wnt signaling pathway. As well, it is a transcriptional coactivator. So these are two of the main functions. So once it binds, it will activate transcription of a certain genes. So it is important and involved in neuromuscular junction defects, as well involved in neural loss associated with AD, Alzheimer's disease, and brain aging. So it's a very important protein, need to be regulated in order to understand how to cure or solve the problems of defects in the neuromuscular junction or deal with the AD and the brain aging. So these are the main functions of spin1. Yes?
What is Wnt signaling pathway?
It's a signaling pathway that occur inside the cell to transmit the signal from the cell membrane to the nucleus through a process of proteins modification.
Involved in what?
Involved in many functions.
Doctor, can I say something?
Okay.
Okay, Wnt pathway, it is involved in cell proliferation. لأنه my thesis is about, like, I studied this pathway. That's why I know this pathway very well. It is related to the cell proliferation. يعني when the cell wants to proliferate, it activates this pathway.
Okay.
Okay. So of course it's not related only to cell proliferation. It has many functions. Okay. But anyway, yes. I don't want to go into details for Wnt signaling pathway because it's not our topic. Continuing talking about info about the spin1 protein. So you can see this figure shows, and you can get it from the internet easily for any protein to know the expression of the gene, if this protein is expressed in a certain specific tissue or cells. You can see, you can get the info, and you can see where the spin1 is expressed, and as well you can see in which tissue? You will notice that Spinone is highly expressed in the brain and spinal cord. You can see even the region have been quantified to know the expression of Spinone. And this explains why this gene is involved in the AD and brain aging, because it's quite a lot expression in the, as we said, in the brain and spinal cord. And for a PhD student, you need to understand very well before working on your protein, if you're doing molecular studies, if already the protein is expressed on your tissue of interest, okay? So we talk now about Spinone. What about H3K4 methylation? What is its main function actually? Okay, so the methylation of the histone, of the lysine 4 of histone 3, it's a differentially mark actively in transcribing genes. So generally it is, once exists, you know that the region of genes around this modification will be transcriptionally active. It's a highly enriched in enhancers. And of course, you know, enhancers is a specific is a sequence of a DNA that is located upstream or downstream of the gene that enhances the transcription of a gene. Normally, H3K4 methylation is highest towards the 5' end of the transcription genes. And this is very obvious because the 5' end of transcribing genes are actually the region of the promoters. So it's a hallmark of the promoters of actively transcribing genes. And they found that it is evolutionarily conserved. What's the meaning of evolutionarily conserved? That means this modification with this function with the same location is present from bacteria till human. Okay, so we talk now about Spinone. We talked about the methylation. We'll understand how the microprotein microarray work. We use protein microarrays to hunt down the methyl binding protein. So in this scenario, for example, you have a slide, glass slide, small glass slide. Okay, and this slide is spotted with methyl binding domain proteins. 98, for example, 98 GST fusion proteins. What are they? Of course, as I said before, Tudor domain binds to the methyl lysine. But it's not only Tudor domain. There are a lot of domains that can bind, a lot of domains. Of course, you know that domain is considered to be a part of the protein, not a subunit. It's a part of the protein. So this domain, we're trying to fish around or hunt which protein can bind to our molecule of interest. Or in another way, we need to know, or we need to hunt for a small molecule that can bind to any of the methyl binding proteins or methyl binding domains of the proteins in order to pick a specific small molecule inhibitor. So initially the slide is spotted by these 98 different methyl binding protein. Of course, one of them is Tudor domain protein. And you lay over them a collection of small molecule inhibitors that is fluorescently tagged. Fluorescently tagged, okay? Soon after washing, you wash the slide and you remove the excess unbound small molecules, and you detect under a machine, of course, that detect fluorescence. You will notice a spot of fluorescence at certain location. What does these spots mean? So you have a single small molecule. You incubate with the slide, and you have the spots here. Can anyone know what these green spots mean?
It means that they are inhibited.
Sorry?
It means that they are inhibited. So they are
they
linked
not
to
inhibited.
the methyl.
Not inhibited. They are.
Doctor, does it? Does it mean? Okay, sorry. Does it mean إنه صار في cross linking between ال added molecules مع molecules اللي كانوا already موجودين على المايكرو array؟
The molecules on the microarray actually they are proteins, domains, proteins.
Yes. So هن هول
Go ahead.
Yes دكتور أنا اللي فهمته إنه الموليكيولز اللي نحن حطيناهم هن في عندهم radioactive substance يعني هن labeled ونحن
fluorescent label.
Yes. Okay so fluorescence label فهن I think هن عملوا binding مع البروتين على الميثايل group. Does it mean that? I'm not sure.
ممتاز. Okay, that's really correct. This is actually what appears under the detector. So you can see down here is all the proteins, okay? And once we pour or incubate this protein with a specific chemical compound, here it says UNC 1215. And this slide was incubated with EML 405. And this slide was incubated with EML 417. You can see that different compounds nearly have either common compounds to bind or different bind to bind. So for instance, the EML 45 and UNC 1215 bound to a protein which is called 53pp1. Both of them bound to the protein, correct? While EML 417 didn't bind. You can see as well, hold on. You can see as well that EML 405 bound to a specific protein, which is spin1. And here is our protein of interest, spin1. So we understood now that EML 45 can bind more specifically to spin1, okay? So this is the real image of a protein microarray. You can see the spin1 here. So let's go further. So we have now 3 compounds, UNC 1215, EML 405, EML 417. Here is the structure difference between EML 405 and EML 417. So let's do some other experiment inside the cells to show the affinity towards the spin1. In this experiment, we overexpressed the spin1 protein in different forms. You know, spin1 protein, it's not only one protein. It's actually a family of protein starting from spin1, spin2A, spin2B, spin3, and spin4. In this experiment, we wanted to pinpoint which spin protein from the family that actually bind with the inhibitor, in this case, EML631. So in this experiment, you can see here, when you pull down spin1 with EML63, you can see the expression of spin1, spin2A, and spin2B, and no levels at all for spin3 and spin4. So the main conclusion of this pull down assay that EML631 can bind with spin1, spin2A, and spin2B. But the perfect binding and the more specific binding evident by the thick band is the spin1, because you can see it's a thick band here, okay? So actually till now, what we know already that some types of these small compounds, small chemicals, can bind to the spin1. We managed to prove that by a protein microarray, by the Hunting method, by the pull-down array, pull-down protocol, and we found that it is have more affinity towards spin1. That's why we continue in the line of spin1. And as well, some structural analysis to identify the region of binding of two examples like EML 405 to Spinone, as you can see from the gray sticks here. These gray sticks is EML 405, and these are the 3D structure of the protein. And as well, we managed to have a structure appearance of the, and the fitting of the pocket, the fitting pocket of EML 631 to the Spinone protein. So let's do some experiment and see if this is Spinone reader ability, because as you know, we said before that Spinone can read the methyl lysine on histone 3. So we have done some pull down assay. So we pull down the protein in a competitive assay. We pull down the protein with a peptide that is already a peptide of H3K4 trimethylate. We increase the concentration of the peptide to see if there is a competitive inhibitor or something in the presence of the 4 different inhibitors. Okay, so you can see here then that upon increasing the concentration of the peptide that have the signal, and with incubation with a Spinone, and in the presence of the different protein, you can see the most affinity goes to, you can see there is a different in affinity between, and there is a difference in competition. So that means Spinone here, this is the signal of the Spinone. For example, let's take this band, you can see Spinone. That means Spinone is bound to the signal, and this inhibitor failed to prevent the binding. But in these cases, you can see there is no Spinone here. That means Spinone couldn't bind to the signal because the presence of the inhibitor at a very high concentration, 50 micromolar, inhibit the binding between the Spinone and its signal. Okay, so what do you think is the best inhibitor from these 4?
EML 633.
632.
Yes, 632.
Yes.
Okay, it's around 632 or 633, okay? Why? Because the smallest concentration, it can bind even at low concentration. Okay, let's go here for another setup inside the cell. So this one is actually in vitro. So all of the 3 components have been put in a test tube. Either the signal, the histone signal, or the protein, the reader, or the molecule, all in the test tube. But here, this GFP Spinone protein has been expressed in cells. And then we'll check with the binding of this protein while treatment of the cells with the same 4 inhibitors. So in this case, you have cells, you incubate the cells with these 4 different inhibitors separately, of course, and then you overexpress Spinone GFP, and you do a Western blot, normal Western blot, to see if you pull down Spinone GFP. Okay, so you can see here that the presence of spin 632 have managed to decrease the amount of Spinone eluted in the Western blotting. Okay, what about 53 pp1 GFP? 53 pp1 is considered in this setup as a positive control, because we know already from before that this EML 632 can inhibit 53 pp1. So use it as a positive control to check if the reaction, if the binding is occurring or the inhibition is occurring. So again, from this setup, you can see that EML 6321 can inhibit the binding of a spin to inhibit the binding of the spin to the reader in the cells. So let's see how this binding or inhibition will affect the activation or the upregulation or downregulation of the gene. Keep in mind, as we said before, that one of the main function of spin1, once it binds to the signal, the methyl signal on lysine 4, histone 3, it acts as a coactivator. So here, after doing RNA-seq analysis, actually RNA-seq basically, you treat the cells and you collect all the RNA, messenger RNA that is formed upon treatment, and then you sequence all these messenger RNA in a single shot, in one shot. And so you will identify, compared to the untreated, if the messenger RNA are upregulated or downregulated. So they found that when they incubate cells with EML631, they found that 933 genes were downregulated. While when they incubate with the spin1 siRNA, what's the spin1 siRNA? Actually, they are downregulating the expression of spin1. In other way, they are stopping the expression of spin1. Okay, so you have as well 936 genes are downregulated. So when you take the combination of both, you will have around 655 genes which are downregulated. Same here, the same principle here, but some genes are found to be upregulated. The intersection of these RNA-seq analysis will show these genes, okay? So now, we already know that some genes are downregulated upon the knockdown of spin1 or the treatment with EML631, as well as there is around 681 genes are upregulated upon the knockdown of siRNA of spin1 and EML or the treatment with EML631. Let's take an example, okay? So RNA-seq can tell us which genes are turned on in a cell and what their level of expression is, and what time they are activated or shut down. So let's take an example. So these are cells untreated, so that's why it's a control. And you can see here, this is actually represent the region of the DNA or the region of the gene of the spin1. So this actually the gene of the spin1 represented by expression. So that's mean you understand now that the gene of spin1 have like three exons, and all of them, of course, because they are transcribed as a messenger RNA, and this is the main result of RNA-seq. So in control, untreated, you can see that spin1 is expressed normally. What if you introduce a spin1 siRNA? You can see that the level of expression has decreased markedly, correct?
Yes.
What about if we treat with EML631? The level as well.
It is downregulated,
It
but
decreased,
not
that much, of course. So from this figure, you understand that spin1 is located on chromosome 9. Are you seeing the pointer of the mouse?
And the
Yes.
cursor, doctor, yes. But it's better if you can use, doctor, the pointer. It's clearer.
So from this figure, you can understand that spin1 gene is located on chromosome 9, and it consists of 3 exons, okay? And it's normally expressed in untreated, while adding of a spin1 siRNA, it will decrease its expression. Adding EML631, it also decreases its expression by around 25%, comparing with the fold change, okay? So that's not experiment, that's a control experiment, all of it, okay? So that's mean, what is the main conclusion of this experiment? Can someone tell me?
Yes, can I?
Okay.
And no, the second part, when the spin 1 is bound to the siRNA, it was inhibited. So the gene for spin 1 was downregulated. So we can see from the graph that it's downregulated. Whereas in the presence of EML-C31, it wasn't inhibited, even though EML is supposed to be an inhibitor, but it didn't affect its regulation.
Okay. Of course, it's not that correct, but you need to understand there is a difference. There is a difference between inhibiting the function of a spin 1 and inhibiting the expression of a spin 1. And totally, both these two are totally different. So we are working on inhibiting the function of spin 1. So spin 1 had to be expressed already. So that's mean here that EML, although we are trying to prove that it inhibits the function of spin 1, but doesn't affect the expression, only by 25% affect the expression. So that's mean spin 1, that's mean that EML631 affected the protein after its translation, not its expression. It's only affect its expression by only 25%, okay? So let's go for, you remember, we have said from the RNAseq analysis that some proteins are upregulated and some proteins are downregulated. So we'll check for one of them, which is interleukin-1 beta, one of the list of the proteins that we have got. This interleukin beta, as you know, is overexpressed and cause a lot of autoinflammatory syndromes. So this is a control expression. So you can see this is the messenger RNA expression of the interleukin-1 beta, and it's located in chromosome 2, consists of 1, 2, 3, 4, 5, 6, 7 exons. And you can see this is the messenger RNA expression of all of them and the sequencing. Once you introduce a spin 1 siRNA, which theoretically it's a different protein, but once you introduce a spin 1 siRNA, you will soon notice a difference in the expression of interleukin-1 beta. Okay, that's mean the level of spin 1 protein in the cell will affect the expression of interleukin beta. And as well, the introduction or the treatment of EML631 will do the same job. So here, it's a direct relationship between spin 1 and interleukin-1 beta. That's mean that spin 1 is involved in the regulation of interleukin-1 beta. Again, let's see some other genes. So you can see here, we have in this experiment, we measure messenger RNA levels in some target genes in liposarcoma. You can see that spin 1 here levels. This is our control setup. You can see why it's control, because we have the blue one is SIR control, and we have the spin 1 knockdown. So in this case, it's the same setup, but we measure for the expression of ribosomal RNA. Interleukin-1 beta, we have just said, another protein, BST2, another protein, it's all different protein. So what do you think the most significant protein that is more affected by knocking down siRNA? By knocking down spin 1 by siRNA, what is the major gene that is affected?
Yes.
Which one?
BST2.
Bravo. So BST2, it's obvious that once BST2, it's obvious when you use red, the knockdown of spin 1, you can notice that the level compared to the control is markedly decreased, okay? So all of these genes are affected actually. This is a control. All of these genes is affected actually, proved by RT-qPCR. Here again, you can see now, of course, in this experiment, there was no inhibitor included, no small molecule included. So the second step, is to include the same setup. Actually, we did the same setup, but we include two types of inhibitor. And you can see in this case, the best protein that is affected by the inhibitor is interleukin 1 beta, correct? And of these two that have been tested, EML631 was the best choice for inhibition of the expression of interleukin 1 beta. So our aim now is, if you have a liposarcoma, in a liposarcoma, normally interleukin 1 as a target gene for SPIN1 is highly active from the control. But if you use either of these inhibitor, especially this one, which is EML631, you will decrease the expression of this interleukin 1 beta, which is very upregulated in liposarcoma. Okay. So, do 631 block spin chromatin association? So what if we need to prove that already SPIN1 cannot bind in vivo on the DNA, on the signal with the inhibitor. So the same setup have been done, and we checked for SPIN1 treatment, SPIN1 overexpression in the cell with the 631 to see if this will affect the overall signal in a chip assay. You remember, it's a chip assay. So I have a protein, activator protein that sits on the promoter of specific genes, and you pull down with the antibody for that protein, the same protocol we took last lecture. So you can see here, in the presence of the inhibitor, the amount of protein you got is markedly decreased. So that's meaning that yes, it is associated with the chromatin, as we said in the last lecture of the protocol of CHIP. So the summary of this work, so initially the protein microarrays can be used to something called target hop, fishing, hunting, and identify the molecules that bind and compete with the domain motif interactions. So we found that a number of compounds are effective in binding with a spin. These compounds, like these 4 compounds, but we still want to know if it's effect only in the binding or it will affect the expression or the function of the spin. So we found that EML631 was active in competitive assay. Remember when we incubated the inhibitor with the signal, methyl signal, and as well in chemoprecipitation experiments, and as well in cell-based coactivator assay like the CHIP one. Finally, EML631 was considered as the most specific SPIN1 inhibitor, and the inhibition of SPIN1 by 631 affected a lot of key genes in cancer cell lines. Okay? And of course, these protocols you have seen and these experiments have been published in Nature Chemical Biology. And I was